Hey everyone, it's Kevin. The structure between a virus and a bacteria is very different. For example, a bacteria has a cell wall and a plasma membrane, and their genetic material is DNA. However, the virus has the genetic material of either DNA or RNA, and it is surrounded by a protein coat. There are more organelles for a bacteria, such as the flagella and pili. But those are optional, they're not in every single type of bacteria. Now let's talk about size. A bacteria ranges from around 0.2 to 5 micrometers, while a virus ranges from 20 to 300 nanometers. The difference is 1 micrometer equals 1,000 nanometers. This shows that the virus is extremely small. As we can see from the diagram, the virus, these are the different types of viruses, are way smaller compared to the whole entire bacteria. Viruses have many shapes, but they're all the same concept. They're just genetic material surrounded by a protein coat. Just like these images. As we can see, this is coronavirus. This is a protein coat. Inside it, you'll find genetic material. Here you find another protein coat, also known as a capsid, and RNA as the genetic material. And in this image right here, you can see that the genetic material would be stored here. Bacteria has a very bad reputation, but actually most kinds of bacteria are helpful. These bacteria are known as flora. These flora are found on your cells. However, some other types of bacteria might try to use your cells as a host cell for food, or they might try to infect your cells using toxins. The virus, however, will try to infect you, and this happens in two types of cycles: the lytic cycle, which I'm going to talk about now, and the lysogenic cycle. In the lytic cycle, the bacteria, the virus, as you can see here, attaches to the cells using the receptors. It then injects the genetic material into the host cell. While the genetic material is inside, it will start replicating. It will then assemble, forming protein coats, and then release, or known as lyse, which will burst the cell and then eventually kill it. Then we have the lysogenic cycle. This cycle is pretty much the same. However, instead of replicating in the cytoplasm, the genetic material will enter right into the DNA and bond with it. So that means that each time the cell replicates, its DNA is hidden inside. And then eventually it will become like the lytic cycle and it will burst and then the viruses will escape. We already know how viruses reproduce, either in the nucleus of the host cell or the cytoplasm. But bacteria can also reproduce, and they reproduce in two main ways: binary fission and conjugation. It's very easy for bacteria to replicate since they have no nucleus. All they have to do is replicate their genetic material, as they did here, split it in half, and then separate. In conjugation, however, this is where bacteria tries to use genetic variation. It will attach to another bacteria using its pili, and then it has a small plasmid. This plasmid will jump over to the other one, get replicated, and be sent back. This causes genetic variation for the bacteria, which will be helpful. Thank you for your attention.